to introduce young academics, we are a new society founded this year based on research in general. So we're trying to marry up students with researchers in any capacity we possibly can. So on one side, it's matching up to obviously research projects themselves. It could also be books, it can be presentations. It's trying to get students authorship in as big as capacity as we possibly can. And a sort of subsection of that society is Research Your Path, which is what we're doing today. And that is trying to get students teaching experience from an early stage. So probably a lot of you know, especially for your medics, for the portfolios, and some of the requirements are to do teaching, including at a national level. I'm running a teaching program, which is what we're doing at the moment. So we're trying to get as many students collaborating with supervisors on a topic that they're interested in themselves. So today we have Lauren and Dr. Banka both talking about clinical genetics, which is Lauren's passion. So we've been running applications for this as well. So we had an application open up for gastroenterology, also another one open up for cardiovascular events as well. So those two have been, we found students for those two as well, and there's supervisors who are very willing to mentor those students. And at the end of this talk, Lauren is going to have an assessment of her teaching from today's session. So that's what Lauren gets out of it, and everyone else gets out of it, is learning something about the specialty itself in terms of the research aspects, which you don't really get at university or medical school. So sort of learning about the clinical challenges, what the current advancements are in that field, and what the future research direction. So if you wanted to go into clinical genomics or whatever, and you don't know where to start, what sort of project you wanted, but you maybe have an idea of maybe a supervisor you want to get in touch with or whatever, but you need to think of an idea to write in that initial cover letter. This is sort of to spark some innovation, spark some ideas so that you know what to look out for, what to talk to when you're networking with professionals in the future in this field. So this is, to give you that complete insight and to answer some of your questions that you may have. So this talk has been structured so that Lauren has reached out to different students, different professionals, to get questions, understanding of what it is exactly they want to understand more about clinical genetics, and they're going to be tackling that head on in this talk, and some sort of Lauren's and Dr. Banka's research as well. So I'm not going to talk any longer, and I'll let you start.
So I'll just introduce our speakers for today. So we have Dr. Banka, who is a consultant clinical geneticist here in Manchester, who's also done a PhD on metabolic disorders. And then we also have Lauren Bradford, who's one of our fourth year medical students, who's done a master's in molecular pathology with a special interest in clinical genetics as well. So I'll let them carry the talk.
Right, should I start now? Okay, great.
Yep, so here we are.
Great, thank you. So can you all see the slide? Yeah, okay. And I think, Lauren, you've got the control, right?
Yeah, I have.
Okay, so thank you very much, Arisha and Komal, for the introduction, and Lauren for engaging me with this very interesting society that you guys have set up. And I think it's really important for medical students to engage in research as early as possible, because there's so much opportunity, there's so much going on. Just expanding your horizons as much as possible during your medical school would be really useful. Now, Komal, I must apologize. The brief that you gave, I think it's probably impossible to cover all of that in 1 hour. And I think the way Lauren and I have tried to structure this is to have maybe more time at the end, maybe half an hour or so, where people can actually ask questions. So rather than me telling you how to go about doing this, might be better if you come up with questions and I can give you suggestions and answers for that. So I'll try my best. I don't know everything, obviously. No one knows everything, but it'll be a good interaction. Actually, I know a couple of people already in this group. So I know there are people who are interested in genetics. So it's nice to see. I know Sharon, and obviously I know Lauren previously. Okay, great. So next slide, please. Great. So really a very broad overview for genetics. So I would say that genetics probably underpins almost every human disorder, if you think about it. So probably not in, let's say, a road traffic accident. Maybe that's one condition where probably genetics does not play that much of a role, but almost any human disease that you think of, there is an element of genetics. So, for example, even right now with ongoing coronavirus COVID-19 pandemic, we know that there's a huge element of not just the viral genome, but actually even the human genome. So the susceptibility to infections is really determined to a large extent by by the variations in the human genome. So really understanding these genetic alteration, whether be it for diagnosis, be it for understanding the mechanism of diseases, be it for predicting risk as to who would get a condition or not. And finally, if you understand all of that, then obviously the management of genetic disorders is or management of disorders is also underpinned by the underlying genetic variation. So here in the picture, you can see a child with cystic fibrosis, for example. There's another child with Duchenne muscular dystrophy. Then you see a couple of brain scans comparing normal versus a brain scan in Huntington's disease. And then, so those are probably quintessential single gene disorders or disorders which are essentially genetic, and there is an element of environmental factors, but less so. And below that, you've got images suggesting disorders which are more probably multifactorial in at least a subset of population. So whether it's dementia or coronary artery disease or breast cancer, for example. But even within these disorders, there would be a subset of patients whose disorder is primarily driven by the underlying genetic variation. So for example, all of you would be aware of BRCA1 and BRCA2 mutations. So although 95% of breast cancer is probably multifactorial, meaning a combination of environmental and genetic factors, in 5% of patients with breast cancer, essentially the disease to a large extent is driven by the single genetic mutation. So really the summary for this slide is that genetic alterations are important and they underpin almost every human disease. Next slide, please. Okay, great. And in this slide, I just wanted to highlight what is a possible pathway for research in genetics or genomics. So, and never the road is sort of as straight as it is meant out, as it is drawn out in this slide, but you would probably first identify the genetic variants. You will then uncover their relationships with the diseases. And then you would try and understand the mechanism. Once you do that, you probably try and develop treatments and management strategies. You will then put them into clinical trials. And then once you get those results, implement the whole knowledge gained from the research into practice. So that's really a pathway, but it most of the times actually doesn't work like that. You are at different points in the different at different points in journey for a particular variation or a genetic disorder and so on. And then there are clues coming from all sides all the time. So, but just to simplify that process, we've drawn this particular pathway. So next slide, please. And I think in the UK, we are really lucky and privileged that genetics and genomics are at the heart of the care that we deliver to the patients, and we really are at the forefront in the world in incorporating genomics into our clinical practice. So I presume that, I don't know how many, 20, 25 people are there in this particular session. Almost certainly not all of you are going to be geneticists, because there are only, I think, 250 or 300 clinical geneticists in the entire country. However, no matter what specialty you choose, be it general practice, be it a surgical specialty or a medical specialty, you would need to understand genetics and genomics in your career. It was always the case, but it will be even more the case going forward in future. So this is really important, and knowing that the system that we work in the NHS really gives a lot of value to genomics and how it helps in patient care. So, so we are really at the forefront, and that also is good for patients, but it also creates a lot of opportunities for us to actually do some world-leading research in the area. So the first, rather the next section for this talk, I'll hand over to Lauren, who will give an example of some of the research that she's been engaged with and how that's helped in those first two steps of this pathway of identification of genetic variants and uncovering their relationships with disease. So over to you, Lauren.
Thanks. So the thing that I've been looking at is improving the diagnosis of genetic diseases caused by rare chromosome deletions. And this sort of started out because there's this clinical need for it. So 1 in 17 individuals have what's termed a rare disease. So while each disease itself is uncommon, when you take them as a whole, it does affect quite a large amount of people. 80% of these rare diseases are actually genetic in origin, and 10% roughly of rare diseases are caused by chromosome aberrations. You can use to identify these chromosome aberrations, but it's not enough to just notice something's wrong with the chromosome. You have to be able to prove that that change to the chromosome is having a negative effect and causing disease. And currently about 33% who undergo this microarray analysis will receive the result of uncertain significance. So we know that something's wrong with the chromosome. It's not as it should be, but we don't, which is obviously difficult for these patients because we want to know what's going on. We want a management plan for them. And that's where the research I've been doing comes in. Survey results from Manchester were collaborated. Greater than 5,000 chromosome variants of uncertain significance were then identified. Over 1,000 of those chromosome deletions that were uncertain significance, and sort of filtered through all of those to find about 250 chromosome deletions, which were likely in critical genes. So genes where there was a high chance of disease being caused. So they were prioritized. And then this thing, so I was going through this list of different genes, different chromosomes, and trying to work out.
Hey bro, what's up?
Had a phenotype that might be linked to it. So the places I was looking at to help with this were different databases. The first one is OMIM, which basically collects different variants or different genes that are linked to known diseases. There's also ClinVar, which is for clinically relevant variations. So if somebody finds a variation in a gene, they can upload it on ClinVar with the evidence and what it might cause. And there's also the Human Gene Mutation Database, which is sort of similar to ClinVar in terms of like people upload variants that they've found. So the whole sort of outcomes from this and the aim of it is to reclassify chromosome deletions of uncertain significance and say, actually, this might be causing the disease, so change it. That then provides a diagnosis to the patient. You can say, this is what's causing the condition. And that has an impact on how you can manage these conditions and treat them, and also look at recurrence risks and assessments for the risk. And the other part of it is you may end up identifying something novel that hasn't been seen before. So I would go through an example that actually turns out quite promising, and it has a long name of MAPK81P3. So started off looking in OMIM for it, and it is actually linked to a neurodevelopmental disorder with or without variable brain abnormalities. So this is good, because if it's in OMIM, it tends to be that it has been linked to this disease now. And what that means is you can go back, and for patients who may have a chromosome deletion in this gene, possibly get them a diagnosis. The next bit was looking on ClinVar for the different variants. So when you have a mutation, there's different variants of that mutation. And from this, you put in the gene you want. It comes up with all the different variants. The ones I chose to focus on were the frame shift, the missense, and the nonsense mutations. And these are because they're the ones that are most likely to be pathogenic, in particular the frame shift and the nonsense ones. And you can see there that quite a few came up for this MAPK81P3. And this was a mix of both likely pathogenic and pathogenic ones. So you're starting to find variants. And the good thing about ClinVar is that people can upload evidence to prove why they think it's likely pathogenic or pathogenic, disease causing. And this is often in the form of papers or Pubmed IDs. So I then went to all the Pubmed IDs that were linked in for this gene to try and get an idea of what kind of phenotypes. So what was the mutation in this gene causing for people? And then you can start building up your phenotype and get an idea for what it might look like. And then you can compare to your patients and see if they share a similar phenotype. So from all of this, the phenotype that I've sort of come up with that seems to be working for this is a range of mental delay. So they're slow in reaching all of their milestones. They sort of walk later in life. They may have some degree of intellectual disability as well. In a sort of anatomical sense, and what can we image, what can we see? There's cerebral atrophy and also hyperplasia of the corpus callosum. And I've got a little picture up there, which is the cerebral atrophy. So where the grey matter isn't as full, it's not there as much, it's shrunk down a bit. They also have gastric issues, the most common of which seems to be constipation, and congenital heart disease is more prevalent, in particular patent ductus arteriosus. And then there's also variable dysmorphism. So dysmorphism tends to be sort of changes or abnormalities in somebody's face and how they look. And you can often sort of notice that that sort of repeats between the phenotype. And people sometimes have similar features. So that's really the work that I've been doing, trying to find out if any of these genes can be reclassified. And yeah, just an example for you at the end.
Thank you so much, Lauren. Sorry to interrupt. Occasionally, your voice interrupts slightly. So,
which bits did you miss?
Oh no, I heard majority of it. It just pauses the minute Zoom catches it up like really quickly. So I think it's maybe your internet connection is slightly unstable. Just to let you know.
Okay. So I listen to it back.
Or another. No problem.
Thank you, Lauren. That was really nice and clear. Did you get problems with my Zoom connection as well, Komal? No. Okay. So I think what Lauren has just shown is really important, because it changes and informs lives of so many patients straight away. So not only does it have clinical impact, it also gives us an opportunity to understand new conditions which may have never been described before, or which may have been described, but very little is known about them. And all of this also informs us about the basic biology of biological functions of various genes and various physiological and pathological processes. So really studying rare disease genetics, and that's what these presentations are really about, is highly useful and informative at the same time. Now, there are obviously lots of different kinds of genetic research. So today, we are both our presentations are focusing on rare disease genetics, but you can similarly apply similar concepts to common disease genetics or multifactorial diseases as well. So I've got another example, which is which would cover the next two steps of the pathway, which is how do you use the information about rare disease genetics to understand the disease mechanisms and then develop treatments and management strategies for that. So next slide, please. So this is actually a work of one of my PhD students who was a doctor, and he specializes in clinical genetics. And then he came over to Manchester from Chile to do a PhD with us. So as part of his work, we identified variants in a gene called EIF5A. And this was done using data from a number of different rare disease genomics projects, one of which was DTT, which is a UK-wide rare disease pediatric genomics project. And we also had Gene Matcher, which is like a website, dating website for geneticists, where you can find if anyone else has a patient with similar mutation or mutation in the same gene as you. And that it helps just to connect people from various parts of the world, not people necessarily, but clinicians and researchers from various parts of the world. So we had a few patients that we identified, and then we used Gene Matcher to identify patients from all across the world. So in the middle there, you can see a diagram, a cartoon for the EIF5A protein, which is encoded by the EIF5A gene. And the lollipops, which are sticking at the top, are the different positions for the mutations that we identified in our research. Next, please. Thank you. So we not only identified that, no, sorry, previous one, Lauren. Thank you. So not only we identified the genetic variants in them, we also collected their clinical phenotype, so the clinical features of these patients. And on the left, you can see the facial features of these patients, and they're, without going into sort of too much technical detail, essentially what it showed was many of these patients had relatively small jaw, which is called micrognathia. We had both males and females in this cohort. Intrauterine growth retardation was seen in about half of these patients. Similarly, neonatal feeding difficulties were also present in some patients. The most consistent feature across our cohort was developmental delay or intellectual disability. And approximately, well, not approximately, but 3 out of 7 patients also had congenital heart anomalies. And the figure on the right is a plot for occipitofrontal circumference, the head circumference, against the ages of these patients. So as you can see, a number of these patients had small head, and some of these were below -2 standard deviation, which is the standard, which is the definition for microcephaly. So we could therefore say that de novo, so de novo means these variants were not present in their parents, but were only present in the children. So they must have occurred for the first time, either in the egg or in the sperm, or very early on in the zygote or the embryo when it was formed. So de novo. Heterozygous means obviously that the variant was present only in one copy of EIF5A, not in both copies. So de novo heterozygous EIF5A variants cause developmental delay, intellectual disability, microcephaly, and micrognathia. So that's the title for this slide, and that's the summary of our findings from this slide. Next, please. So once we had identified this new genetic condition, which had never been described before, we next wanted to understand what are these variants doing. So what are these variants doing to EIF5A function? So the one thing which is very interesting about EIF5A is that it is really a very, very important gene, and that is reflected in its extreme evolutionary conservation across species, such that the EIF5A gene is also present in yeast. So yeast, as you all know, is an organism, but it's kind of very far away separated from human beings. But not only that EIF5A is present in yeast, actually the sequence of the amino acids that make EIF5A is very similar in humans and in yeast. So that means that if you change that sequence in any way, it is likely to be deleterious to the function of EIF5A. And therefore, across millions of years of evolution, the gene structure or the protein sequence of that gene has not really altered. So therefore, what we could do is that we could introduce certain the mutations that we had identified in our human beings, in our patients, introduce them into yeast and see what those mutations might be doing to yeast. So of course, in yeast, you cannot have micrognathia and microcephaly as you read out phenotypes. So in yeast, what we first tested was whether yeast with these particular mutations that we'd identified in our patients, did they grow well, just like your normal wild type yeast. So the diagram at the bottom left is just a schematic showing how we produce those variants. And on the bottom right, that shows that yeast with the mutations did not grow as well as the wild type. So the whiter the circle in that particular figure, the better the growth is. So as you can see in the wild type column, in the column with wild type, the circle is nice and white. And then just below that, T48N is a mutation and the circle is a bit grayish, and similar to that in 106R as well. So that proved to us in an in vitro assay that the EIF5A variants that we had identified in our patients, they seem to impair EIF5A function. Next, please. So this raised the next question for us as to what is the basis of deleteriousness of these missense EIF5A variants. Next, please. So to be able to really answer that question, we need to know what the normal function for EIF5A is. So EIF5A is important in translation. So translation means that you've got your mRNA. So from DNA, RNA is transcribed, and then from RNA, proteins are translated. And it's that translation in which EIF5A is really important. And it's not important in all translation of all proteins all of the time, but it is specifically important for proteins which contain the amino acid proline, and not just one proline, but chains of proline. So as you can see on the right side, that you've got your ribosome, which is happily synthesizing a particular protein, but then there is a sequence where there are 3 prolines which come one after the other. And then because the structure of proline, the biochemical structure of the amino acid, the ribosomes stall because they cannot deal with multiple prolines in a sequence. And that's when EIF5A comes in and helps the ribosome to progress. Otherwise, the protein synthesis would stall. So it's really quite important in terms of a synthesis of especially complex proteins. Please. So using this information, and I won't go into the details of this particular slide, but we did an assay which looks at polysomes. So essentially what it is trying to see is how are the ribosomes arranged during protein synthesis. And what we found, and we don't really have the time to go through the details, but what we found is that if you use a mutant yeast, then the profile of how the ribosomes are functioning, it changes. And the interaction between the mRNA and the ribosomes, between EI5A and ribosome, that is altered as well. So that gave us an insight into how EI5A mutations might be causing the disease as they might be causing. So that raised the next question for us, which was, next, please. So we thought, okay, now that we understand to some extent what the mechanism is, can we do anything to find treatments? So can we rescue these phenotypes in our model systems? So for that, next, please. We knew from the previous work that has been done that, next, please. Thank you. So we knew from the previous work that's been done that there is a group of substances which are called polyamines, and within that a specific type of polyamine which is called spermidine can actually help in the function of EIF5A. So we thought, okay, if you have a system where EIF5A is not working well, can we actually supplement that system with spermidine and see what happens? So when we did that in our yeast systems, and I didn't show a lot of data in other slides, but it's, you probably just have to believe me right now that when we treated these yeast with spermidine, we saw that the phenotypes could be rescued. So that was encouraging, but yeast is yeast. But we wanted to, and therefore we wanted to test this in a higher animal to see whether is it just restricted to yeast, or can it actually help maybe humans as well. So for that, next please, we generated a zebrafish model for the disease. So we impaired EIF5A in zebrafish because, again, just as I said, it's highly evolutionarily conserved. Zebrafish EIF5A is very similar to human EIF5A, and when you impair EIF5A in zebrafish, you see similar problems like you see in patients with EIF5A as well. And when we treated these zebrafish with spermidine, we could rescue their phenotypes as well, which raises hope that perhaps this treatment can be used in humans as well. And spermidine is a non-toxic substance which can actually be used like a food supplement almost. So that's really exciting because next please. So in our conclusions, we could say that we've discovered EIF5A variants as a cause of a novel human developmental disorder, and that these variants impair EIF5A function through that lead to impairment between the interaction of EIF5A and ribosomes, and that these impaired functions can be recovered by using spermidine as a and that raises a treatment possibility. So this was published quite recently in a journal called Nature Communications, and there's obviously we're very grateful for all the patients who participated in that and our funding agencies as well. I think that's the last slide. Lauren, maybe could you press next and see whether there's anything else? So yeah, that just sort of brings us back to our pathway that we started with. We've left the last two bits, clinical trials and implementing knowledge into practice, because I guess that's almost common to each and every specialty that you would talk about. So hopefully we've given you some taster to the first 4 steps of this pathway. And I hope that has stimulated you to think of some questions, which is going to be our next slide, Q&A. So I'll shut up now, and then I guess the floor is open to everyone to ask any questions.
So I wonder if it would be easiest for people to type questions in the chat, and then we can read them out.
Enable is like how to get into genetics.
Sorry, Chloe, did you ask a question? I don't think I could hear it properly.
I think her question was around how to get into genetics.
How to get into genetics. Okay. So can I just ask, is everyone here a medical student, or are there people who are from non-medical backgrounds as well? Yeah, okay, so we've got more medics and some non-medics as well. So I'll try and cover both the streams. So for medics, I guess the straightforward way is to apply for a clinical genetics training position, which opens at ST3 level. So as you would know, that you would need to do your foundation year 1, foundation year 2, ST1 and ST2. There are no genetics jobs in FY1 to ST2. So the first job that you would be eligible for would be at ST3 level. So to get into genetics, you would need membership of either sort of medicine or pediatrics, or literally any patient-facing specialty. You can do your MRCP, MRCOG, MRCPCH, or whatever, MRCGP as well. And then you can apply for ST3. So that's one way to get into genetics, which is the traditional route. Then there's also you can apply for an academic clinical fellow position. So these are academic training pathways. Academic training pathways in which you can you get time reserved for research, and you can also get time for clinical for your clinical training as well. But again, that also starts at ST3. Although the ACFs can start earlier, so it might be that your ACF starts at ST1, and then you end up doing ST1, ST2 in some other specialty and coming to genetics at ST3 level, or in some cases, ACF might start later as well. So really, if you wanted to get into genetics, you need to apply for it at ST3 level. Of course, how do you prepare to your CV to get into genetics is probably another question. And if anyone wants to know that, I can answer that later. But I just wanted to cover Mona and Zohaib's questions as well, because they're non-medics, and how can you get into genetics? So I think broadly speaking, again, this may not be the only way to get into genetics, and again, genetics is quite broad. There's plant genetics, there's animal genetics, and so on. But I'm specifically talking about clinical genetics or genomics. So there are probably 2 broad pathways to do that. So one is that you can be a research scientist. So you can do a PhD, for example, in a related subject. which is related to clinical genetics, and that way you can sort of carve out a career for yourself, investigating either the mechanism or genomics or treatments, etc., and so on. So that's really one way to do it. The other possibility could be that, for example, if you wanted to work in an NHS genetics or genomics lab, then there is what's called a scientist training program. So if you Google NHS STP, scientist training program, you'll find loads of information. There is a national intake every year for STPs, which is a 3-year training program, which trains you to be a registered clinical scientist. But then there is a long way, as with any medical career, of doing FRC path and lots of other exams, etc. So you train and you work and you give exams at the same time, but that is how you become a clinical scientist. So you can be a research scientist or you can be a clinical scientist working in the NHS lab. And then there are people who probably combine the two as well. So for example, I'm a clinician scientist. So I look after patients with genetic disorders, but I also spend half of my time doing research on genetic disorders. Similarly, there might be basic scientists who spend time in the NHS lab, providing diagnoses to patients, but they might also spend time in the university research lab, doing research on genetics. So there are so many different models, etc., to do this. I hope that that wasn't brief at all, but it probably answers some of the questions.
So we have another question, which is how much of it is hands-on and how much is practical work?
So I think, Zoe, you are a biomedical person. So how much of it is hands-on / practical work? So it really depends on what work one is talking about. So, and what do you mean by hands-on? So if by hands-on, if you mean working in the lab for a lab scientist, then it would be to a large extent, it would be hands-on work. It might be doing the lab work yourself, but it might also involve doing the analysis of the results that are generated from the machines, and so on. So in fact, that's also quite hands-on, I would say, if you're sort of doing like what Lauren presented, doing database searches, reading papers, extracting, synthesizing the evidence, and utilizing that to generate reports and so on. So I would say a lot of it is hands-on, but I don't know whether your question was a bit more specific about certain types of hands-on or practical work.
And then there's one on if you want to do both clinical and research science, does this mean you have to do an STP and a PhD?
So if you wanted to do research, substantial research as a basic scientist, PhD would be essential. I think even without a PhD, you can engage in research like anyone working in the NHS, for example. It is possible. But if you really wanted research to be a big part of your career, then even whether you're a medic or you're a non-medic, doing a PhD is a very good idea. I would say that even if you're not planning to do research, doing a PhD is a good idea because it means that you get training in not just in training in understanding how evidence is generated, how research is done, and that really can be helpful in your day-to-day job, non-research job as well. So yes, the answer to being a research scientist, you have to do a PhD. For being a clinical scientist, most people will undergo STP training programs. Right now, there are some avenues where you can be a registered clinical scientist in sort of a parallel ad hoc manner without going through an STP. I don't really know how things will be in future, but as we know more and more, these career pathways are getting formalized. So I would say probably thinking about an STP program would be better because it's usually well structured and you get a lot of sort of proper training and opportunities to do projects, etc. So, so Mona, in response to your question, it is a yes. And it is not a bad thing, actually, it's quite good. And it might seem a bit daunting, but actually it's fantastic training, both for STP and PhDs. So next question is from Navia, who's asking, describe the role of a clinical scientist and how does it differ from a research scientist in an academic setting? So Nadia, I guess you can unmute yourself because I just want to know your background. So you asking from are you a medic or a non-medic?
I'm a non-medic, I do biomed.
Biomed, okay, yeah. So really the role of a clinical, I'm not a clinical scientist, I'm a medical clinician scientist. So I'm probably not the best person to answer that question, but I interact with loads of clinical scientists in our department. So I know to a large extent what their day-to-day jobs look like. So really clinical scientists are, so all the lab data results that are generated, clinical scientists would analyze those results, synthesize the evidence, and produce reports. And then that's sort of the main job. But of course, the more experienced ones would be involved in setting up new assays, new tests, in a lot of managing the teams, and so on. So it's really a role of a medical, rather a hospital lab scientist, a bit like what you might think of in, let's say, in biochemistry, what a clinical scientist does in biochemistry, similar sort of a role in genetics. It does differ substantially from a research scientist in an academic setting, because research scientists would be mostly thinking about research questions. They would be generating preliminary pilot data, then writing grant applications, doing those experiments to find new knowledge, and then writing research papers and reporting that to the wider community through papers, presentations, conferences, etc., and so on. So I think, and that all some of these roles are overlapping, because obviously clinical scientists can also be engaged in research and writing papers and writing grants and doing research, etc. But the fundamental focus probably of clinical scientists would be diagnostics, whereas for research scientists the fundamental focus would be research. So that's how the two roles differ from each other.
And then we have another question, which is, what advice would you give to students wanting to gain research experience early on, i.e. in year 2?
Yeah, so Chloe, I think you are medic, so I'll answer that question from your perspective. So in Manchester especially, there are lots of opportunities throughout your medical school to get involved in projects. So you have PEPs and APEPs and QPEPs, for example. So, for example, I think the most substantial official research project that you get to do is APEP, and I think it's in year 3, probably year 3 or year 4. I cannot remember now, but you get to do a
Yes.
6-week project in APEP, and if you're interested in genetics, look for APEP projects which are either based in the genetics department. So the genetics department in Manchester is the largest department in the country. So if someone is interested in genetics, Manchester is absolutely the right place to be. And usually in APEPs, our department will have several projects available for students. If you're interested, you can even reach out to people in our department to flag your interest and so on. So a bit like maybe how Lauren has done. So Lauren's project is not, strictly speaking, sort of part of your routine work, routine medical school work, but she's interested and she emailed me, we had a meeting, and we sort of involved Lauren in an ongoing project, and she's done absolutely amazingly well. And I hope that she's had an opportunity to learn something from us as well, as she's engaged with the project. So really, there are these sort of set pathways that you can follow. But I think there's no substitute to actually reaching out to people, sharing your interest, sharing your ideas. And I guess if you talk to 10 people, probably 5 or 6 times nothing comes out of it. But really you need just 1 or 2 of those meetings or ideas or things to work for things to actually have an impact on your future career. So my advice would be just find out what you're interested in, and then find out people around you who share that interest with you. I don't know if that answers your question or confuses you more.
Then I'd sort of second all of that, in that if there's something you're interested in, do reach out to people. And sometimes, you know, you'll sort of get to know one person, and then through them they know somebody else, and that can sometimes be a way in as well. And yeah, just sort of be proactive with it, put yourself out there, and there are some really good opportunities, and I've really enjoyed working on this. So yeah, it's good.
And one more thing which I can probably add to this is lots of students nowadays also do intercalated degrees. So if you, for example, did an MRes as part of your intercalated degree, then you will have an opportunity to do a substantial research project as well. Even in MSCs, you will get a smaller project as well. So yeah, you could also think about doing that. In addition, when you're on your clinical placements, you could also, for example, look out for opportunities to maybe write case reports. So case reports would be would require less amount of work. It's not like a research project altogether, but still it can be something really interesting and valuable. And then you can get a publication out of it that will again add to your CV when you're applying for your specialist positions. So there are lots of ways in which you can demonstrate your research interest and interest in a particular specialty.
I got a question. So I am really interested in using CRISPR as a strategy to treat human disease. And I'm just wondering, is it considered as a clinical scientist or a research scientist? And also another question is, at the moment, are there a lot of research about CRISPR in human disease, especially in Manchester? Thank you.
Yeah, okay. So the first question that you asked, Mona, was about whether using CRISPR as an application for treating human diseases. So currently that would be very much in the area of research and less so for a clinical scientist. Now, maybe in future when it becomes more routine, then people who understand the technology, have worked with that technology, maybe that will become part of sort of maybe not diagnostics, but therapeutics of the mainstream, which might be in the hospitals, which might be in biotech companies, pharma companies, and so on. So I think the answer to your first question, right now, it is very much within the research area, although the progress is astonishing and it's quite fast. The second question was about how much of that is happening in Manchester. So a lot, really. So when you're using CRISPR, there are a couple of, broadly speaking, one is that you can use CRISPR to understand disease mechanisms. And once you understand disease mechanisms, you may not necessarily need to use CRISPR to treat something. So for example, like the EI5A example, which I gave, if you understand the mechanism, then you can use some other treatments rather than gene therapy, for example, or CRISPR-Cas mediated gene therapy. So there's a lot of work going on in various model organisms, in human cell models, etc., where we are using CRISPR-Cas techniques to understand the diseases and impacts of genetic variants. That's one. And secondly, there is a lot of work going on in using CRISPR-Cas for gene therapy as well. But that's a bit behind because, of course, with CRISPR-Cas, there's lots of off-target effects as well. And if you're wanting to do something to, let's say, give cells, CRISPR-Cas treated cells to a human being, then you want to be sure that off-target effects are minimized as far as possible. So, so right now, a lot of CRISPR-Cas application is around understanding mechanisms and actually even delivering some diagnostics as well. But therapeutics is not far behind, and a lot of this work is happening in Manchester as well. It is obviously one of the sort of leading biomedical universities in the world. So, we do have all the sort of cutting edge technology and facilities to enable this kind of research.
Thank you. I would like to have a follow-up question. So is it possible for non-medic students to apply some summer research experience in the Genomic Center?
Yes.
All right, thank you.
So our Manchester Center for Genomic Medicine, as I said, is one of the largest clinical genetics departments in the country, probably in the world even. And actually, we have about 300 people working in our department, and out of that, only 30 are medics. A vast majority, obviously, are either clinical scientists working in the lab or genetic counselors who work in clinics who are not medically trained, but they're trained in conveying and assessing risks associated with genetic disorders. So I don't think we touched that aspect anywhere in last 1 hour, but that's another avenue to get involved in clinical genetics for non-medical people. If you're not wanting to work in the lab, more in the clinic, then one can think about genetic counseling as a career option as well. Okay, are there any other questions?
I think that might be all of them. Oh, I've got one which is, do you interview students for research positions?
Yeah, so any research position, especially the paid research positions or paid, well, for example, PhD studentships, etc. Usually there'll be an interview process to go through. Sometimes it might be that you just reach out to a supervisor and you say, okay, I want to work in the lab over the summer for a couple of months or so. So that there won't be like a formal interview process for that where multiple candidates are being interviewed. But of course, you'll need to contact the supervisor, have a chat with them. They'll want to know what your background is, what your previous experience is, what skills do you have, what is your interest. And then if there is an ongoing project, then they might be able to fit. So there will be a process of meeting up and discussing rather than a formal interview. But usually for paid positions, of course, there would be interviews involved. What do I look for in undergraduate candidates? Any tips? I might throw that question back to you. What do you think would anyone look at? But we're 2 minutes over anyway, so I'll answer that question in a straightforward manner as far as possible. I think one would look for genuine interest. So it's not something that you're just doing because you think it's a good thing to do, or so a genuine interest in the field, maybe some prior experience or something in the CV that shows that, yeah, you're genuinely interested in this field, that you're enthusiastic. If you've got some skills which are relevant to the project or the field, then that's really good. And yeah, I mean, in general, that someone is asking interesting questions, right questions, and so on. So I think it's really, really hard to say what do we look for in undergraduate candidates. So when Lauren approached us, we thought that she'd actually worked with one of my collaborators previously. So that showed me that she was genuinely interested. And then she was trying to do something which is not sort of part of her routine medical school work. So that means that someone will do that only if they're really, really interested, keen in that. You won't sort of spoil your evenings otherwise, isn't it? So that's that sort of thing. I think it's really hard to say what we look for. But yeah, commitment, enthusiasm, and interest is what we would look for, I think.
I think we've come to the end of all our questions there. Thank you so much, Dr. Banka and Lauren, for this amazing talk. And sorry for the mistake, guys, as well. I've sent a second link out for the feedback form, so you can